Well, thank you, everybody. We're running a little bit late, so try to make it as interesting. And relevant as possible. I'll cover a little bit of what Klaus has covered in that video, and again, he'll answer questions directly by email. But if you do have any questions on his particular presentation, I might be able to help there. It's always a pleasure to come to Spain. I thank Juan and his team. They're always incredibly hospitable. They always organise beautiful weather for me, which is helpful. And I will do my best to speak slowly, so if you have any questions, it is supposed to be a workshop, so please interrupt at any time. So, my first question, who would like to work for Klaus Wiemer? As you can tell by the way he showed about making dishes, he's very particular about how he runs his labs. And he's very modest, of course, and says we're quite happy with a 50, 60, 70% pregnancy rate. I mean, who wouldn't be? So I think I take from what he presented that really now in Europe we have no choice but to use global LP if we want to use global media. The LGPS system, which was brought in from the very beginning, is no longer available in Europe. It will never get a CE mark because of the nature of how it's produced. So we had to find an alternative. And fortunately, the data that we've heard earlier, and hopefully what Klaus has presented, has convinced you at least that LP works as well as LGPS, and hopefully will work as well in your labs with any other media that you potentially want to use. I would also like to thank the presenters from this morning for putting my heart in the right place and not giving me any palpitations. My role within the medical affairs department and at CooperSurgical is one where we want to get the best out of the media and every other piece of equipment or consumable that we manufacture. And it's part of our role to make sure that you guys get the best out of it. And so when you have to sit and listen to people presenting on what they've used of yours, you hope that it's going to give you good results. And mostly because we certainly don't want to do any harm to any of the patients that have obviously been using these media and many other things that we produce. So thankfully everything seemed okay. It's exactly what I expected to hear. It's never going to be universally better than what people currently use in an environment where it's always going to have a bias against it, because people are using a media, whatever that might be, and this is the test media, and so consequently they've had a lot of experience with one and very little with another, and so you always expect a little bit. And I can show you in the presentation some presentations or some publications, and they rarely show huge differences in outcome because they're not powered to. And even Klaus has said in his study, the numbers are not there to say one way or another whether one's better than the other. They can basically say more or less they're the same, but we'd need many, many more patients to go through treatment and go through this protocol before we could say one was better than the other. So ultimately, this is what I'm hopefully going to cover. As I say, I will speed up a little bit because it's getting towards 4 o'clock and I can tell people have filled their brains. So, global media. Global media, be it culture media, fertilization media, whatever, has been designed to be a family of media. This was where the principles came from at the beginning. So whichever one of them that you use, they all have the same chemical backbone, and they only change in their formulation what's needed for that particular function. So, for example, fertilization media needs higher glucose concentration for sperm survival and cumulus cell health. So it has higher glucose concentration than the culture media, but everything else is the same. And it's the same for the others. Whatever their function is, HEPES replaces bicarbonate, but everything else is the same. And we want to do it for one reason alone, is that we want to keep the embryos stress-free throughout their development, and this seems to help. And we know that that's the case. But I do want to reiterate before I get into the media that anything that we use in the lab is part of a media or a culture system, and the culture media is only one part. And if we don't get the other parts right, the culture media cannot perform to its optimum. And so this is just some of the things that we might think about that could impact our treatment and our success. And teamwork is obviously one of them. And again, one of the good things about coming to Spain is that the embryologists I meet in Spain are all generally very well informed and have good labs and good success rates. That's not the case worldwide, and you'd be surprised and horrified at some of the things that I do see, and one of these days I may write a memoir and share them all with you. But teamwork's incredibly important. Who would like to work in a lab doing 3,000 cycles of IVF per month? Okay, so we now have several clinics in China doing 35,000 cycles per year. I don't know anybody in Europe that would want to work in that environment. There is no teamwork, it's a factory, there's no quality control. How they don't mix up gametes is very hard to say. So, we're lucky in Europe, we have strong regulation here, so we have to do things the right way. And so these are some of the practical aspects. Quality control is extremely important, and again, if we don't get this right to maintain things on a regular basis as the way they should be, we will never get the best results. Okay, so bear that in mind. We'll talk a little bit about it later. You heard this morning, my colleagues did a great job in explaining some of this to you in Spanish, so you've heard it in Spanish and now in English. So the two philosophies that have come around about designing media are either you use a single-step media, i.e. let the embryo choose what it wants from its environment, or you use something that you design based on observation. Now, actually, if you look at defined media for anything in science, it will tell you that you shouldn't use physiological observations to determine it because they are variable, and you should actually use just observation to determine what the optimal media is. And so you can make your own judgment, but we know they both have their limitations. The sequential media had its limitations by the variability of observations in the various things that they measured. And single-step media, so simplex optimization media as it was, has its limitations because it was done in a mouse model. We can't do this in the human, so we had to extrapolate from the mouse. And as everybody knows, mouse and humans are not necessarily the same. Some people could argue differently, but we are not the same, and we have some distinct advantages, not least of which, if you put cleavage stage embryos back in any other species other than the human, you get pregnancies. So thank God for that, otherwise we wouldn't all be sitting here today working in this field. So again, that approach leads us to the single step or the sequential media, and again, I don't deny at all most of the commercially available media. If you use it at its optimal, you will get some background results. We know that. Whether you get optimal results depends on many other factors. So the questions that we ask, about the concentrations of the components that we saw, this is really only is biased against sequential media, but as I said, single-step media does have its drawbacks in how it was developed. But ultimately, is anything normal, natural? And I like this next little cartoon because it tells you everything you need to know about what we do in the lab. So on the left is a very dynamic in vivo environment. The embryo development is not in a big pool of liquid. It's in very close contact with the walls of the fallopian tube. It has interactions with all of that. The analogy that we use on the right, that what we do in the lab, is putting a pea in a swimming pool. Okay, this is not normal. And so either sequential or single-step media are never going to be normal. They're very far from it. So we need to bear that in mind. We can't mimic what happens in vivo. Whether we ever will be able to, whether microfluidics will come along at some point and help, who knows? But at the moment, we're a long way. So I don't think you can say sequential or single-step media are normal. They're a long way from what you see in vivo. So bear that in mind as well. So the history of global. And Klaus's name's on here in 2002. So it started off as simplex optimization media. John Biggers, who was the doyen of culture media, was involved from the very beginning. It became KSOM for human work, so increased potassium concentration. Then Klaus and John Biggers introduced amino acids. If you put the same concentration of amino acids you put into animal cell culture, human embryos die. So they thought, well, what should we do? And they literally just halved the concentration of amino acids, essential and non-essential, and suddenly human embryos are quite happy. Then they changed the glutamine amino acid to a dipeptide, which became the initial global, and it became alanine glutamine, and then it was changed to glycyl glutamine, and that remains the same today. So culture media itself is not actually that complicated. Some of you have probably heard this, when I started as an embryologist, I was told in my training that the culture media was the most important thing that we had in the lab, and therefore it would be the last thing I was taught to make. And when I was actually shown how to make media, I was horrified. We had a number of bottles that we lined up. We added water, salt solutions, protein, antibiotics. We inverted the bottle a couple of times, and then the embryologist held it up to the light. And I said, "What are you doing?" He said, "I'm checking the pH." I said, "What?" I said, "This is a solution we're going to inject into the patients." He said, "Your eyes are very, very sensitive to colour, and this is the way we've always done it." So I said to him, "So what if the pH is incorrect? What do we do?" "Oh, well, we have 1 molar sodium hydroxide, and we have 1 molar hydrochloric acid, and depending on whether it's too acid or too basic, we add them dropwise. So as you all can imagine, if you want it at 12 o'clock, you start here. So you add 1 and you go past. So then you add the other and you go back the other way, and you end up, so that was bottle number 1. So bottle number 2 then doesn't need any alteration." And so I said, "So we've made 10 bottles of media. They're all different because they've all had different amounts of things added to them. And we're going to inject these into a patient, and we've checked the pH by eye." And he said, "Yeah, but that's the way we've always done it." I said, "So if somebody from outside came to the lab and asked us how we made the media, you'd be happy to tell them that one of the critical things that we do, we just hold it up and say, well, it's about 7.2. Amazing. And we've come a long way from that. Unfortunately, the hangover from it is that now, should anything untoward happen in the embryology lab, the media gets the blame. So if your results dip at all, it's always who made it, or where's the media, who's it, what lot is it, where did it come from? It must be the media. And unfortunately, we know nowadays it's probably not, but it still gets the blame. So it's a very simple solution. Salts, a buffer, some energy sources, amino acids, EDTA for chelation, and that's 1 dipeptide to stop ammonia production, antibiotic, and phenol red as a pH indicator. And then just to reiterate, this is the global family. And I like this slide because it doesn't matter whether it's global LP, global HP, LGPS, all applies the same. So the protein concentration in each of those will be different. But if you look down, the sodium chloride concentration across every product in the range is exactly the same. The potassium chloride is the same. And then it only varies where it needs to. So where you look at buffer, where it needs to work in a CO2 environment, bicarbonate, where it needs to work outside the incubator, HEPs. That's as simple as that. So we try to make it so that when the embryos are moved from one solution to another, they are as stress-free as possible. So again, just to reiterate, they're all the same. Remarkably, in the very beginning, we used to use BSA, bovine serum albumin, in human embryology, would you believe? Fortunately, we don't anymore. So, also mentioned this morning, oil is absolutely critical. The number of times I talk to embryologists about why they're cutting down on the amount of oil that they use to save a little bit of money. And as we've moved from doing day 2 transfers, which is what I started with, where we hid a lot of the errors that were going to occur in the culture media, we've now moved to day 5, day 6. Oil has become even more critical. If you don't use enough oil or use too smaller drops over the time of culture, you will change the environment that the embryos are in without doubt. And the other thing, again, is if people take account of the viscosity of the oil, this has an impact on the equilibration time for the dishes. You need at least 12 hours, even with the light oil or paraffin oil. And if you get to lifeguard oil, which is very viscous, then you need even longer. Klaus said 19 hours. He's always at the extreme of everything. 19 hours is a long time, but it indicates to get fully equilibrated dishes, we need to make sure they're in the incubator for a long time. So this is, again, just to illustrate that, and this is very nicely indicated, because if we look at our drop, this is what I see embryologists doing. I see them holding the dishes up and looking. I say, what are you doing? Oh, I'm checking the oil is covering the drop. And say, but if you do it that way, so it's literally just covering, you will end up, without doubt, in extended culture with loss of water, and therefore you'll have increases in the salt concentrations, the pH will change, the osmolality will change, everything. So drop size is important, oil is very important, and as we heard earlier, mostly because of volatile organic compounds. Not all of us work in the best labs in terms of air quality, and that again varies around the world. So we need to make sure that we use enough oil and our drops are big enough so that we have a good volume to surface area ratio, and again we don't get evaporation. Jason Swain did a paper, his name was mentioned a few times this morning, where he did different types of dish making, and as Klaus did with the GPS dishes, he recommends that you put drops in your dish, you put oil over the top, then you take the drops out and you put new media in, and this way you minimize any changes that you might get. It's a pain in the neck to do it, particularly if you have a busy clinic making dishes that way, but that's the way he recommends to have the least change over time. So a single step media can be used to culture from zygote to blastocyst without question. There's enough data out there, but you need to be careful that you have a good system in your lab to allow that to happen. And changing media, whether it be the same media or a different media as in sequential on day 3, is a stress on the embryo, and we need to minimize it as much as possible, as is opening and closing the incubator doors. So if you're going to introduce any media into your lab, this is for global, but it could be any, you need to have a plan of how you're going to do it and make sure that you do it right so that nobody is compromised in any way. And really we can't get a really conclusive answer in the time frame that is commercially viable. So no, I don't believe any company is going to be able to come to an embryologist who says, yes, I want to trial your media and I need to do 1,000 patients in your arm with your media and 1,000 patients with the media we currently use, and that will help me decide. It's just not feasible to do that. So we recommend initially 30 to 40 patients with the test media, gives an indication of whether it's going to work in your system. And then beyond that, we can talk about how we refine it. But you can never get a truly positive answer in terms of significant results with a relatively small sample number. It's recommended that you do a prospective randomized trial. Some places do, and it was nice to hear somebody did this morning. Most places don't do that. They do an evaluation rather than a trial, partly because if you call it a trial in many countries, you then have to get ethical approval, and that again adds to the complication of doing it. So we talked about optimal culture conditions. I stress time and time again to people, so mostly they listen, sometimes they don't. Every media is different. So you cannot assume that a test media will work in exactly the same way as your current media. And the main consideration for that is bicarbonate concentration. And we had a discussion over lunch between Sage 1-step and Global. Now, Sage 1-step has bicarbonate concentration about 50 millimoles per litre, and Global has about 19.5. So at the same CO2 concentration in the same incubator, they will not give you the same pH. And therefore, depending on what the optimal pH is, they may or may not be working at their best, and you need to bear that in mind. And this graph on the right shows different media, different CO2 concentrations, and different pHs. And as you can see, they're almost all different. So even in the same environment, you do get differences, and you need to bear that in mind. So we recommend doing sibling oocyte trials or sibling zygote trials. Again, some people will do it, some people won't, depending on their workload. But in some way, you need to assess whether one media is going to work better in your hands than the one that you currently use. And certainly, as far as Global is concerned, over the time period that we've had it in the market, we have worked out what the optimal pH is. If you ask any other commercial company what the optimal pH of their media should be, they'll tell you between 7.2 and 7.4. Now, we know for global that that's not true. It will work within that range, but if you want it to work optimally, it should be below 7.3 and should ideally be somewhere between 7.25 and 7.29. Now, that's a much smaller range than 7.2 to 7.4, and it has its implications. So you need to test it in your system. You need to make sure that you use an incubator dedicated to the trial media or the evaluating media, and it should be the same as one that you're testing against. I was in a lab last week where the CO2 concentration was set the same for each of their incubators in the lab, including a Jerry, some benchtop, some box incubators, and every one gave different pH readings, even though they were all set at the same CO2. So it is incubator dependent as well, and environment and everything else does play a part. So bear that in mind. If you're going to compare two media, they should be given equal opportunity to work at their best and not compromise them. Again, the worst thing that can happen, and this has happened in some quite famous labs, they decide on two media. One works at relatively high pH and one works at relatively low pH, so they choose somewhere in the middle. And now all that does is it compromises both. So you'll not get good results from one or the other, and that doesn't tell us anything at the end of the day. And what we're trying to do is optimize our system so that we get as many patients to take home a healthy baby as we possibly can. That's our ultimate goal, and that's what we're striving to do. Do you need to refresh? And again, Jason Swain did a paper saying, here are some of the potential problems. And as I said, when I started at the beginning of the 90s, we knew our culture systems were compromised. They were nowhere near optimal. But because we did embryo transfer on day 2, much of its inadequacies were hidden. So we didn't culture any longer, so we didn't have time to cause too much chaos. If we went back to those times and said we should do day 5 transfers, we'd have a nightmare, and a majority of patients wouldn't get a transfer because we were not good enough at the time, and we didn't recognize some of these issues, particularly around VOCs, particularly about evaporation, pH control, and all these sort of things. As I say, they just weren't in our forefront of our minds. We wanted to get patients pregnant at any cost, and we put 3 embryos back in most, and still the majority didn't get pregnant. So we've come a long way, and if you believe, which I truly do, those results that Klaus has presented, we've come a long way from where we were. I always hark back to the original Louise Brown. If it hadn't been for Bob Edwards' stubbornness, Louise Brown was born on their 104th attempt. So not with her parents, of course, but with other parents around the time. If he hadn't truly believed that it would work, most people would have given up by then. So we've come, I say, a significant way along the pathway. So can it be used with time-lapse? Of course, this is what brought about the big expanse in single-step media. So even those that didn't really believe it early on thought that actually if you've got time-lapse and you can observe embryos without disturbing them, then wouldn't it be great if we had a media that could support that development? And so we've seen a growth in single-step media from that point onwards, whereas global for many years was on its own and stood out from the crowd. So again, it can work, it does work, and again, some of the research that's been done will show you can do it uninterrupted. The question, I don't think we need to answer this, does it work as well as sequential media? Then I think the answer is obvious, it does. There's some early papers, and again, they don't show significance in some of the numbers that are relatively low, so the number of patients for pregnancy rate, for implantation rate, but where you had more numbers, i.e. in this case in blastocyst formation, then you do get a significantly increased number, whereas the others are not significant. And that's more or less what we saw across the board initially. And that's what people were looking at. Was it the same, or would it give us the same results? And if so, it was beneficial then to use it because it had some other advantages. If you use a single-step media as opposed to a sequential media, your life suddenly becomes a little bit easier. And again, in labs that are very busy, stressed, anything that can cut down workload, potential to make mistakes is obviously going to be beneficial. So even though these things didn't show particular benefits one against the other, I expect if we looked at utilization rate as well, that would be significantly better. But people moved because if it wasn't any worse, why use 2 media when you can use 1? Makes our QC and everything else that much easier. So just another one that shows exactly the same thing. So just to reiterate, global media works as well as any other. It seems to be one of the most robust media on the market. We've now shown that whether it be LGPS or LP, it works the same. Many of the clinics that used it initially in the UK before the CE marking issue raised its ugly head, didn't use HSA at all. They use dextran and other macromolecules, and it works just as well with those. So it's a very robust media that gives you great results, and like all media, only if you use it optimally. That's what we're here for, the Medical Affairs Department within CooperSurgical. We've been to some of the labs of people here. It's an open invitation for me to come to your lab if you need it. You know, we can help, and our true goal is to make sure that if you use any products from CooperSurgical, that you use them to their best advantage, and therefore we get patients, as many of them, taking home healthy babies as possible. Okay, there's a few more slides, but I think I'm going to leave it there. I think that's the message I want to take home. Don't be afraid of the media. Don't be afraid of single-step media, but please do take care that you try as far as possible to optimize your conditions. If you do that, then I'm absolutely certain you'll get great results, and we've got a good team around you to help you do that. So thank you for your time, and any questions, please feel free.